Okay, good afternoon. It's 1:30, so we're going to get started. Good afternoon, everyone, and welcome to the Office of the Chief Scientist Science Seminar Series. For those of you who don't know me, my name is Vanessa Swarbrick, and I'm a Senior Science Advisor with the OCS and your host for this winter's seminar series. On behalf of our entire team, welcome to today's event. I would like to start today by respectfully acknowledging that in Alberta, we are on the traditional gathering places and home to the peoples of Treaty 4, Treaty 6, Treaty 7, Treaty 8, Treaty 10, and the Métis Nation. I would like to acknowledge the diverse Indigenous people whose footsteps have shaped this territory and whose histories, languages, and cultures continue to influence our communities today. Wherever you find yourself, I encourage you to recognize and acknowledge the relationship that First Nations, Inuit, and Métis have with the land upon which we all live, work, and learn. In keeping with the tradition of gathering, these science seminars provide an opportunity for us to get together, to exchange knowledge on interesting science happening across Alberta in an open learning and sharing environment. The hope is that through our dialogue today, we are all able to improve our awareness and understanding and to put this new knowledge into practice to support evidence-informed decision-making wherever that occurs. As you may have noticed when you joined the call, we are recording today's seminar. Your microphone and video are disabled. However, you can enter your questions or comments at any time during or after the presentation via the chat. The chat is not being recorded. After the presentation has concluded, I will read out the questions in the order that they were received. And with that, I'd like to introduce today's speaker. Today, we are welcoming Lisa Wilkinson, who is presenting on enhancing resilience of Alberta's bats to a deadly disease. And she'll be taking us through some of this white nose syndrome information in just a moment. As the species at risk biologist, Lisa has worked with a range of taxa from mosses to salamanders. As threats to bats have intensified over the past 2 decades, her work has increasingly focused on bat conservation. Lisa chairs the Provincial Bat Working Group, coordinates provincial monitoring and recovery efforts, represents Alberta on national and North American bat working groups, and develops and delivers outreach about bats. And with that, I'm going to pass the floor over to Lisa.
Yeah, thank you, Vanessa, and thank you for this opportunity to talk about white nose syndrome. It's an important issue, a devastating disease that has killed millions of bats throughout North America. And this talk about the probiotic is, I consider it a little ray of hope in an otherwise dark story. And I would like to acknowledge my co-author, Corey Olson with the Wildlife Conservation Society Canada, who has been instrumental in all of the work that we've been doing. I'll begin by a quick overview of Alberta bats and explain more about what white nose syndrome is, how it works, and then I'll lead into the probiotic treatment and our current and future plans. Alberta is home to at least 9 species of bats. They're all, of course, little insect-eating bats. And I think that the role of bats in pest control has never been more valued and understood as it is now, because there's such a paucity of bats on the landscape. People are really noticing they're missing that pest control. The farmers in the south, it was estimated that bats were saving them billions of dollars in pesticides. So we're definitely missing our insect-eating bats. So white nose syndrome is a disease of the winter. And bats in Alberta have different strategies to deal with the winter when there are no insects around. We have 3 species that actually migrate. So these species are not vulnerable to white nose syndrome. We've got a red bat, hoary bat, and the silver-haired bat, and they go southern US. We don't have a good idea where they all go. Some of them are probably slowly making their way back north to Alberta right now, but these species don't hibernate. So the bats that my clicker, there we go. Oh, sorry, I just skipped a bunch of slides. There we go. Sorry. Yeah, so the 6 other species that we have hibernate. And the thing is, there's still a lot of mysteries about bats. We don't know where most of our bats hibernate in all of the Western North America. We don't have as many large cave systems as they do in the East, where they could have, or previously had tens of thousands of bats hibernating. They probably hibernate in cracks and crevices in rocky areas, riparian areas. And they do, in fact, hibernate in caves. But in Alberta, we only know of a handful of caves where these bats hibernate. And they're really important places. We know from one cave in particular, and banded bats, that they'll travel up to 300 kilometers to get to these hibernation sites. So they're very limiting, they're very important. Without knowing what many of them are, it makes it harder for us to really understand, the impact of white nose syndrome to some degree and how to protect these sites. All the caves that we know of are protected. People are not allowed to enter them during the winter, and some are closed off all year long. So white nose syndrome is caused by a cold-loving fungus called Pseudogymnascus destructans, which I will call PD for brevity. So PD, and I'm going to talk a little bit more about it, is this fungus, and it really likes to grow at 5 to 10 degrees. It has quite a range, and so it is found in caves and other places that bats hibernate. And there's a really important distinction here between PD and white nose syndrome. So PD can be in the environment but not cause white nose syndrome. Small amounts will not irritate bats. When larger loads of the fungus are present in the environment, it'll begin to penetrate the tissues of the bats where they don't have any fur, wings, nose, tail, and then it triggers a physiological response, and basically the bats just arouse and they quickly use up all their energy stores because there's no food, it's winter, and they starve to death. So in terms of monitoring and reporting, there is an important distinction between PD and white nose syndrome. So in brief, our hibernating bats travel. Again, we don't know exactly where they're all going. One of the things we do suspect is that they have mating swarms in the fall. So this is an opportunity where males and females will come together. They'll mate. Females have a delayed fertilization, so they wait till the spring before they continue on with reproduction. And this is a time when bats, if they go into a cave or hibernation site that has white nose syndrome, has PD, they can then pass it on to other bats, and then they mix and mingle and move to other hibernation sites. So it is primarily spread bat to bat. So in the winter, probably early winter, everything's fine. But the longer the bats are in there, if there's a lot of fungus in the cave, it'll eventually start to penetrate those tissues, and some bats will arouse and die. By spring, hopefully there'll be some survivors coming out, and they will retain signs of white nose syndrome for maybe 30 days. After that, because their immune system is no longer suppressed, their metabolism is going fully, they're not in a cold environment where the fungus thrives, they can recover. And if you catch the bats in the spring, it looks like everything's fine, but then they return to these infected hibernation sites again in the fall and have to run the gauntlet all over again. So that spring period is particularly important for us as a time that we can, since we don't know where most of our bats are hibernating, this is a time when we can try to catch bats and look for signs of white nose syndrome. And just to let you know, so if you haven't heard about how PD got to our shores, we suspect it was on some gear or clothing from a caver from Europe and China. This is it's native. The bats do not die from white nose syndrome. They probably evolved with it, or there was some kind of period when bats first started to decline the populations. Eventually, those that were able to survive with resistance, and now it doesn't affect them anymore. Our bats, this is a naive population. They didn't develop with this fungus, and so it's new and very deadly. It landed on our shores in New York in about 2006, 2007. And to date, 12 bat species have been diagnosed with white nose syndrome. So you can see it's really spread all the way across the continent. And if we zoom in and look a little closer to Alberta, and the colored sections are where we've detected PD. Now I'm switching to PD, the fungus. And so between 2022 and 2025, we've gone from the south to the north of the spread of the fungus in Alberta. So it does spread very rapidly. It's not surprising because it's what we've seen in the east, but it is nonetheless very worrying. You'll see that in BC, they've only had one detection of PD a few years ago, and they haven't had any since. So detection of PD and subsequently white nose syndrome depends on how jurisdictions are surveying and monitoring for the disease and whether they're able to detect it or not. Unfortunately, 2 weeks ago, cave hibernacula in Alberta was surveyed, and we found that white nose syndrome is now here. Again, it's not a surprise, but it is devastating. So we know the disease has gone from a toehold to a foothold in Alberta. So it's more important than ever that we take steps to try to reduce the impacts of white nose syndrome. And also a couple of weeks ago, a paper was just published that tells us that PD is more prevalent than currently recognized, and it's evolving and diversifying. So research is really going to have to stay current with how this fungus is changing and growing, and it's it spreads very easily and rapidly. So how do we detect it? So we first detected it in southern Alberta in 2022, as I mentioned. And our primary method for detecting the fungus is through bat guano. And we've been collecting it at bridges. So while not many bats actually roost during the day under bridges because they tend to be cold, many bats will just hang out temporarily in bridges at night, and they leave guano for us to collect. And from that, through qPCR analysis, we can confirm the presence of the fungus. And if we have caught bats, again, it's that early period before they've shed the fungus, we can take wing swabs and similarly send them off for qPCR analysis. But this, the guano is really our prime detection, our early warning system, and the one that has revealed all our PD detections from the north to the south. And guano has a secondary purpose that a different DNA analysis can reveal species. So that's also very helpful for a cryptic nocturnal species that aren't easy to study. So our first detection for PD was 2022. 2 years later, not surprisingly, we detected white nose. And in this image, I'll explain it to you. It's under UV light, and the orange spots are areas where the fungal hyphae have penetrated the tissues of the wing. And for some reason, they fluoresce orange. So to the naked eye, this bat looked fine. But when we looked under the UV light, we were able to detect this. And we knew it was the early days of starting to detect white nose syndrome. This bat clearly wasn't in bad shape, but we knew it had been exposed. So the following year, we went to the same site. And as you can see, all the membranes now are really covered from PD. And in the lower images, you can see wing damage from that fungal penetration. Most of the holes will mend themselves, but they're obviously coming out of hibernation in not a very good state, and there's contraction of the wing membrane, and we don't think that's going to change. The other thing to consider is these are the survivors, right? Remember, I said we don't know where most of our bats hibernate, so there could be a lot of bats that perished during hibernation. These are the ones that didn't get a really heavy load or right at the end and were able to come out. And hopefully these bats will be able to replenish their energy sources because bugs are around, but it's questionable whether females would have the energy to follow through with reproduction when they're coming out so weak. And for biologists who have gone into caves to look for bats and check for white nose syndrome, that's the only place you see that diagnostic white muzzle. It's not visible once they've left their hibernation sites and come out in the spring, and bats die. And typically they will be flying out of the cave in the winter because they're hungry. They need to find food. But I said I wanted to talk about hope, and so how we can help our bats. So it's been nearly 20 years since white nose has this tidal wave of the fungus and consequently white nose has come across our continent. So as you can imagine, there's been considerable research. What can we do to help these bats? There's no magic bullet yet, no silver bullet. We can't eradicate the fungus. We can't cure or prevent the disease at this point in time. All types of mitigation are just kind of localized helping hands to reduce the impact of white nose. So the more bats that we can get through surviving that difficult hibernation period and then reproducing, the better. Now, we know because it's been almost 20 years, there are some signs of hope. In 2020 was the first evidence of some genetic resistance. So we know some bats do have a natural resistance. But keep in mind that 90 to 95% or more populations in the East have been wiped out from white nose syndrome. So that's a staggering decline. And these species typically only have one pup per year. So recovery is decades long. So it's hard to come back from that. So this is positive. And again, we just want to provide more of a helping hand through mitigation. This is from 2025 in Wisconsin. They'd actually noticed their third year of an increase at one site. So that's more positive news. Some sites aren't seeing recovery, some are seeing stabilization, and it takes a long time. So what are we doing to help these bats? There's a myriad of ways and trials that have been going on. Fungicides and UV light are two ways that can destroy the fungus. However, they're limited, and there are places that it wouldn't be advisable to apply a fungicide to a cave ecosystem, for example. And if you recall that earlier picture I showed you of a cave, how vast it was, you can imagine the impracticality and unlikelihood of covering those surfaces with fungus-killing UV light or fungicide. It just wouldn't work. So another very promising treatment is oral vaccine. It's being trialed in the States right now, and it does seem to be effective, but you have to have bats caught in hand to be able to treat them, and bats aren't the easiest to catch. So the probiotic cocktail is the one that I'm going to be talking to you about today. And in this image, this is a bat just receiving water. So those bats we were catching in May that were in rough shape were dehydrated and very readily taking water. So this goes back to 2017. There is a consortium of researchers that had the idea to isolate naturally occurring bacteria that are found in the environment, like soil, and sometimes on bat wings. And in the end, they found 4 strains that worked together really well to repel or reduce the fungus. So in that Petri dish, the happy face is that the 4 beneficial or probiotic bacteria, they've been challenged with PD, and you can see the clear circle around them. There's hardly any fungal growth there versus farther out. So this was a really tremendous and encouraging finding. Their next step was to develop a prophylaxis to reduce fungal activity on bats in the winter, so less chance of them getting white nose syndrome. So their challenge was, how would they get this cocktail onto the wings of bats? And this work is being started in BC and then also in Washington. So they needed to be able to reach a lot of bats. And I will say right now, people have asked me, can you go into a hibernation site and treat the bats? And first of all, if you recall, we don't know where most of our bats are hibernating, so that's not even an option. And when bats are in their hibernating state, they're at a very fragile state with low metabolism and immune system, and it's not advisable to treat them with this. So the next best thing is to find out where bats are raising their young, called maternity colonies. Females tend to gather in largish groups, and the places that we know about are man-made structures, buildings or bat houses. So by tracking and identifying these maternity roosts was the first step to applying the probiotic. So this is an image of my co-author at our study site. And so BC started out with the field trials first, and they've gone through lots of iterations of how to apply it. Initially, they tried the live bacteria. We use a freeze-dried bacteria that we rehydrate 30 minutes before we apply it. And they've gone through a lot of trials of different types of sprayers and other permutations to figure out how best to deliver it. But the idea is, when the bats are out flying at night, you spray the inside of the bat houses or an entrance to a roost. The bats will go in, it'll coat their wings. Ideally, it becomes part of their wing microbiome, and then they spread it to other bats. And that is really important. So as I said, these trials started in BC, and a couple of years ago, they went into Washington. Sorry, I know this map is a little bit blurry, but they have treatment sites where the bats receive the probiotic, and then control sites where they were not receiving it. So the results are still a little preliminary. The work's going on, and don't have time to get into the details. In general, the bats at the treatment sites, there's a greater percentage that have one or more of those probiotic strains, and the amount of the probiotic is greater. We can actually measure it by number of cells per centimeter square in the surface area of a bat wing, which is something we're going to be doing in Alberta as well, to be checking that. So that's all very positive news. And probably what's even more encouraging is all the control sites. So bats were not, those bat roosts were not sprayed with the probiotic, but bats being recaptured there do have probiotic on their wings. So that suggests that the bats that were treated, this probiotic is becoming incorporated into that microbiome, and then they're leaving these sites, probably going to wintering sites, mixing with other bats, and that in turn is being incorporated into the wing microbiome. So the wing-to-wing, bat-to-bat transfer is probably the most important part of this experiment. Now, as I mentioned, BC only has one incident of PD. It's not in their study site. So we can only look at treatment versus control for PD and white nose in Washington. And so they have found at their treatment sites, the incidence of PD is lower. The amount of PD that they're finding on the wings is lower. And even, as I said, those probiotic control sites, that applies to the Washington sites as well. And that's only been going on for 2 years. So that's very encouraging. And they have not had any white nose develop at their treatment sites. Sadly, they have white nose at their control sites, and one of them might be on its way to blinking out. But this is all very positive news. So here in Alberta, for many years, we've been watching this research, talking to the primary researchers who are doing it, thinking that if we are going to apply some kind of mitigation to Alberta, this seemed the most promising for a number of reasons. And remember, I was saying there are lots of limitations going from, say, a UV treatment in the lab to the real world. What can we do? How can we best reach the bats to help them? And we thought this was a good idea. And so last year, after in May, we were catching those bats with really more advanced white nose syndrome. We decided it was time to act, but we had to know, are the beneficial bacteria in Alberta? And fortunately, we had taken wing swabs in previous years. And with our partner at McMaster University, we confirmed that those probiotic strains were present in Alberta. And there are other places, other jurisdictions like Ontario that are also starting to do these tests as well. And we're finding that most of these probiotic strains are fairly prevalent. So that's great news. And just to qualify, the fact that it's in the environment, these bacteria, they may or may not be on a bat's wings. And if it is on a bat wing, it may not be in sufficient quantity or combination of the right kind of bacteria to repel the fungus. So that's what we're doing, is introducing it where it isn't already on the wing and enhancing it where it already is on the wing. So we targeted July to start our first treatment of the probiotic, and at the group of bat biologists who've been working on this, we also talked with a couple of our scientists in EPA before we went ahead with this. And I'll explain why we chose July. So this is still research in progress, learning about what are the best times to apply the probiotic. The treatment sites have been applying it twice a year. This year, we're going to be starting in May, and that's that window of time post-hibernation when we can catch the bats and see if they've had white nose syndrome. And then we're going to go back in July. And the reason we're doing it in July is we want to spray the bat houses when there aren't any bats inside, namely pups that can't fly. So while the probiotic wouldn't be detrimental to the pups on their own, we didn't want to spray them with a wet solution because from a thermoregulatory point of view, that wouldn't be very good. So based on a lot of the work my co-author has done, he had a good idea of when the pups were going to be volant. And so that's why we targeted a certain time in July and to spray them then so the pups would be out flying. If you wait too late into the summer, the colonies begin to move around more and disband. So it's a very narrow window. And again, this year coming up, we're going to do May and go for that period in July as well. And we don't know really how long does the probiotic last on substrates. They're still working on that research from the trials in BC and Washington. We did swab these bat houses in September, but we haven't received any of the results for that yet. And just the image there, you can see, well, one person is treating the bat houses. We have a team that is catching bats in mist nets so we can handle them, evaluate their health, etc., species, reproductive condition. So we have a team that really a number of people who've been working on this for several years now. So very coordinated efforts to measure the bats. That's a forearm that's being measured. We take their weight. We take swabs for PD. We're also swabbing to do a different analysis for that to figure out what that wing microbiome is. And we're going to be doing that a lot more in this upcoming year. So what is it before the probiotic is introduced and what is it after? So the study site, which I kind of consider it ground zero because this is where we've had all our first detections of white nose syndrome, is Emerson Bridge Park, located in the southeast of Alberta. And Corey has been tracking this site for a number of years. There are 13 bat boxes. And if you'll recall the earlier image, with a strong flashlight, you can see up into a bat box and see how many bats are in there to count them. You can also count bats that are leaving at night. And admittedly, it's not a precise metric because the bats move around. But if you do enough counts at the right times of year, we get a fairly good idea, and it is one of the metrics that we rely on to look at changes in local bat populations. So in July, we successfully sprayed all the bat houses in one night. However, this is the number of bats we counted relative to 2 years ago. So from over 2,000 to just over 200. So that's obviously not good news. The little bit of good news is we did catch at least one young of the year bat. So they had reproduced. A couple of the females were reproductive. So gives us a little bit of hope for that site. But we know this location has been hit hard. We don't know where these bats are hibernating, but obviously their hibernation sites have a lot of PD in it. The other site is the DNA Gardens. It's closer to Red Deer. They have 6 bat boxes that have been monitored for many years. We got to all those in one night. Good news is the total number of bats in those 6 bat boxes hasn't changed much. The bad news, unfortunately, there's always a good and bad news scenario in this story. If you look at the wing, you can see a bit of a scar on it. So that could have been a bat that experienced white nose syndrome, had a tear in the wing, but the wing mended. And again, so this bat was a survivor. So I'd like to consider that a little bit of good news. So next, going forward, we're going to continue application at our 2025 treatment sites. As I've said, we're going to go back in May and then in July, and we're going to add hopefully 7 other sites. This is a rough map of the sites we're looking at, with X being where the sites are going to be treated, and then the check marks are going to be our control. So we're going to be doing something similar to the other research that's already gone on. So we'll have our treatment and control sites. We'll be handling the bats, we'll be treating and we'll be swabbing the substrates. And we're also, as I said, going to be getting a lot more baseline data on that Alberta bat wing microbiome. And we're going to be applying it to at least one bridge maternity roost. So in the southeast of Alberta, some of those bridges are warm enough to actually support females raising their young. So that hasn't been tried before, and we're not sure if that's going to be a substrate that the probiotic bacteria will last longer on. Not really sure. We're going to develop a scientific advisory board. As I mentioned, we have consulted with our EPA scientists, and hopefully they'll continue to work with us, and as well as some of the other folks doing some of the lab work and other research to help guide us as we go along. And so I just wanted to also mention that this is part of a very comprehensive recovery or bat monitoring and recovery effort. We have a draft recovery plan, and this is a simple infographic to show you how many types of methods we need to use to try to study bats, because they are very difficult to study. So the middle column there shows you whether the method is for trying to get at population size and distribution, or as symbolized by those green blobs, it's disease. And as I've already mentioned, whether it's PD or white nose, it can be very different what we're able to learn. So just in brief, so acoustics, I'm guessing many people are familiar with ARUs, automated recording units that we put out. We use ones that are for ultrasonic calls, and they're great at picking up bats, not so good at distinguishing species. There are limitations, but we're doing this as part of a whole North American coordinated effort called the North American Bat Monitoring System. We've been doing it for over a decade now. So this is really important to look at, to some extent, species of bats, but just overall our activity levels changing that could indicate bat numbers are changing. I've already talked at length about our the bridges and how helpful the guano is for both species as well as PD detection. It's really been a wonderful tool for bat biologists is the guano at bridges. We can also collect it at bat houses. Bridges in particular have been helpful. Misnetting, talked a little bit about that. That's an important opportunity to get our hands on bats, to look at their health, reproductive condition, and in the spring, if it looks like they're coming out of hibernation, having survived white nose syndrome. I also talked about those maternity roost exit counts, and I cited them in the Emerson Bridge area park where we were doing our surveys. We also have a lot of citizen science who have bats in buildings or bat houses on their property, and they monitor those for us. Over the years, many of them have collected guano. They've mailed me guano and I've sent it off to the lab. So we've been able to ID it to species, and they're also able to do their counts. And so it's really helpful. And that's how we can get a little bit of idea on some of those local bat populations. And finally, hibernacula are really important. And again, I've talked about that, how it's limiting because we don't know where a lot of them are, but they are, of course, very important. So we tend to use a whole suite of methods to survey the bats to try to get an idea, both on their population trends, distribution, as well as the spread of the disease and impacts of white nose syndrome. So finally, I would just like to acknowledge all the researchers who put time into this, who did all the work in the years before us that allowed us to basically hit the ground running. So we could start the research last year and start applying the probiotic. And notably, Alberta is the only jurisdiction outside of those study areas that have started to apply the probiotic. I know the Northwest Territories is looking into it as well. And so other jurisdictions are paying attention, but first they have to make sure they have the naturally occurring bacteria in their environment first. But it is a very promising avenue of treatment. There's a number of people who have helped with bat work over the years. This is just a small list of the folks who helped last year. And I'd also like to acknowledge the original caregivers of this land. And with that, I'd like to take questions. I wanted to leave a lot of time for questions. From my experience, there are a lot of good questions and thought-provoking questions that come out of this work. Nobody knows all the answers yet, but again, it is a little bit of positive news in an otherwise fairly dark story. So thank you for your time.
Thank you so much, Lisa. We do have a ton of questions that are coming in, and I just want to say before I transition, I really enjoyed your talk. It gosh, it really strikes a chord thinking about how do we manage species that maybe we don't see as often when we're urban dwellers, but how fundamentally they're important to managing our ecosystems and how we have to pay attention to what's happening around us.
Yeah, that's a good point. And I think a lot of people didn't appreciate them till they started to disappear.
Yeah. With that, I'm going to wrap up our seminar today. I want to thank you so much, Lisa, for your presentation. It was so informative, and I think we're all going to go home today thinking a lot about how to support some of these creatures around us that we don't see maybe all the time.
Yeah, thank you.
Our next seminar for folks online is going to be on March 24th. We're going to get a presentation from Dr. Craig Emerton here from EPA, who's going to be presenting on a network approach for river monitoring and management, which is a case study for diverse and changing North Saskatchewan river basin conditions. If you're not on our mailing list and would like to be added, please follow the link that my colleague Kristen is just adding to the chat right now. And with that, I'd like to thank Lisa again for sharing her work with us, to all of you who tuned in, and we hope to see you again soon.